Hello, and welcome to Biology EOC Review Session 1. I am Mrs. Spencer, and today I'm going to go over biochemistry in cells. So first we're going to start by talking about what makes up life. Since biology is the study of life, we need to understand everything that is important in the composition of life. So the matter, or the four main compounds that are found within living things, are carbohydrates, lipids, proteins, and nucleic acids. These are all considered organic compounds, which means that they have carbon and at least one hydrogen atom in them. So carbohydrates are things like your breads, pastas, and sugars. Lipids, things like fats, oils, and waxes. Proteins, your bones, hair, skin, enzymes, and nucleic acids are things like DNA. So first let's start by talking about carbohydrates. All carbohydrates are going to have the elements carbon, hydrogen, and oxygen. The word carbohydrate even kind of tells you this. Carb stands for the carbon element, hyd stands for the hydrogen part, and then the drate means that there's oxygen with it. The actual building block of a carbohydrate is a monosaccharide, or single sugar. Mono means one, saccharide means sugar. So these are the smallest little compounds or smallest pieces that build up big carbohydrates. Some examples of carbohydrates that you might be familiar with are things like glucose. That is the form of carbohydrate that all cells are going to get from your food that you eat in order to make energy. Sucrose, this is table sugar. Cellulose, this is a very common carbohydrate found in plants. Lactose, a carbohydrate that's found in milk and some people are actually allergic to. Starch is another main carbohydrate. This is a complex carbohydrate that actually hooks a lot of single sugars together for storage and is very common in plants. Glucose, as I mentioned in a little bit earlier, is the energy sugar. That is the sugar found in that all food that you intake is going to be finally broken down into, and the actual molecular or chemical formula for glucose is C6H12O6, and we will be hearing about glucose much more as we move through these different review sessions. I also want to point out that I've highlighted the last three letters of the different kinds of sugars that were listed here: glucose, sucrose, cellulose, and lactose. Notice that they all end with that suffix of -ose, and that is a common thing for sugars. So a lot of times when you see that -ose at the end of a word, that indicates to you that it is a type of sugar. Next is lipids. This is another complex organic compound, and lipids also have carbon, hydrogen, and oxygen, so those same three elements that are found in carbohydrates. Lipids are going to be things like fats, oils, waxes, blubber, and they are primarily made up of fatty acids. That is the building block of a lipid, are fatty acids and glycerol. And lipids are long-term storage of energy. Glucose and the other carbohydrates are our primary source of energy, but when we need longer storage of energy, we turn to fats and lipids. So lipids help with long-term energy, and they also help with protection. Fats can provide protection and insulation for living things. Next we have proteins. Proteins are going to also have that carbon, hydrogen, and oxygen that we've been talking about. But we're also going to add nitrogen. The element nitrogen is going to be found in proteins. Proteins has an N at the end, so N for nitrogen is the additional element found here. The building block of a protein is an amino acid. Those are the smaller parts that make up larger proteins. And proteins kind of make up everything about an organism. They are ultimately what the organism is made up of. So examples are things like collagen, which is found in your skin and your bones, Hemoglobin, which is a protein found in blood that helps hold on to oxygen. Microfilaments, these are small proteins that are found in muscles and cells that help with structure and muscle contraction. And a big one that is very, very important is the enzyme. Enzymes are different kinds of proteins that are going to help with increasing the speed of chemical reactions, basically helping our body and the organism to work and do the functions necessary. So proteins have a wide variety of jobs. We don't like to use proteins for energy. Sometimes we do in very rare cases, but when proteins are used as energy, then that ultimately means you're using yourself, you're breaking your body down for energy. So it is a last resort for energy. Proteins are mainly involved in making up the organism as to what they are, the structure. All right, and then the last major organic compound in living things is the nucleic acid. And the nucleic acid has the carbon, the hydrogen, the oxygen, and the nitrogen like the protein, but now we're adding a phosphorus, so it has an additional element known as phosphorus. And the building block of the nucleic acid is going to be a nucleotide. Okay, and nucleotides, again, are the smaller pieces that make up the nucleic acid. And some major examples of nucleic acid, some of the big complex molecules, are things like DNA, which are like the genetic blueprints for an organism, and then RNA, which is basically kind of like the worker. They help build the organism based off the directions given in DNA. So those are the four main organic compounds found in living things. Again, we will be talking a lot more in detail about the glucose, carbohydrate, the proteins and how those are made, and then of course also genetic information with the DNA and nucleic acids as we move through these different sessions. But for now, this is the main things to remember about the different organic compounds. Another major compound that is very important to life is water. Water makes up approximately 2/3 of every living thing. So it is a very important part of living things. It is not considered organic. It is inorganic because although it has hydrogen, it does not have carbon. So it is an inorganic compound, but that doesn't make it any less important to life. There are four major things about water that make it so essential and important to living things. The first is that it is considered the universal solvent, which means that it easily dissolves most substances in it. So water is pretty much the base of most solutions. Your blood is an example. Blood is mostly water. It is the base of the blood. And so when we have very little water, when we get dehydrated, our blood cannot flow like it's supposed to. Another property of water that makes it very essential is its ability of cohesion. Water molecules are polar, which means that they have a slight charge to either end, and this charge allows for the water molecules to kind of stick together. So they kind of stick to one another, and when they do that, they build like a surface tension. And this is what allows us to have basically kind of like a thin membrane over the top of water. So you can have water bugs walking on top of water. When you drop water onto the ground or the counter or something, it forms droplets. When it comes out of the sky, it's the form of drops. When you look into a tube or a cylinder or a glass, it tends to have a curved surface at the top, and that is because it's kind of sticking to the sides of that container. And when you did your lab with us, the one where you had to drop the drops on the penny, a lot of you remarked how you were surprised at how many drops could fit on the penny, and that is because of this cohesion of it sticking to itself and sticking together so it wouldn't overflow. So that is really important, and this is what helps to allow, again, like I said, the surface tension, and as well as allow for tall trees and plants to get water up to the very tip tops at the very tall parts of the tree and the plant against gravity. So this cohesion helps. Expansion is another very important property of water, and this is the fact that when water freezes, when it becomes a solid, it actually expands. This is not normally what happens when a liquid becomes solid. Most liquids, when they become solids, shrink because the molecules tend to come together. But in water molecules actually spread apart, and again, this is because of that polarity, that charge on either side of the molecule kind of pushing away from one another in these cases when the water freezes. And because it expands, it becomes less dense than the actual liquid water, so that allows it to float, which is why when you put ice into a soft drink or a Kool-Aid or just another glass of water, it will float because it's less dense. This is extremely important in our cold areas of the world. This allows for when we get freezing temperatures for the ice to form on top and allow for the lower areas of the water to remain water and not freeze through, so that the wildlife and marine life that live in those areas do not die every time we get cold winters. And lastly, water is very good at moderating temperature. Water is extremely slow to heat and slow to cool. So if you think about this, think about your pools right now. It is springtime, becoming summertime. We've had some 90-degree weather, 80-degree weather. However, if you've gone to the pool recently, you might have noticed the water temperature is still a little chilly. And that is because even though our air temperature has heated up very quickly to 80, 90 degrees with these warm, spring weather, it has actually, it takes much longer for the water temperature to increase as well. So the water temperature is going to increase at a much slower rate. It's going to take a little longer for it to get to a more comfortable warm temperature for the summer and springtime. And this is actually really important because, again, since the majority of living things are made up of water, it helps keep us from boiling when we do get 100-degree weather outside, and it helps keep us from freezing when we get freezing weather outside, so we don't just, the water in our bodies doesn't just fluctuate spontaneously with the temperature outside. So it's actually a good thing. All right, so now that we know a little bit about the compounds and matter that make up living things, let's talk about the cell. The cell is the basic unit of life. It is the smallest part of a living thing that actually carries out all of the characteristics of life. Do remember that in order to be considered living, a living thing must be able to reproduce, must be able to grow and develop, must be able to adapt or respond to its environment, must be able to be organized and have actual, organization, and must use energy. Okay, so if something has those 5 characteristics, then it would be considered a living thing. If it does not, then it's not considered a living thing. So the cell is the smallest unit that actually does have all of those 5 characteristics. And there was, basically some ideas that we have come to understand that are common about cells, and that this is lumped together in an idea known as the cell theory. So the cell theory states that all living things are composed of one or more cells, and this is where that organization part comes in of what living things must have. All cells must come from other cells, which makes sense because if a cell is a living thing, it has to be able to reproduce, and that's where the cells come from other cells are. And then cells are the smallest unit of life is the last part of the cell theory, which just states that it does show all the characteristics of life. So those are the 3 main points to the cell theory. Now, how do we know this? Well, we know this because of the scientific method and because of microscopes. Because scientists used the scientific method and did experimentation, and because of the actual invention of microscopes allowing us to be able to see microscopic things such as cells, which are too small to see with the naked eye, we were able to find out these things about cells and know that it is considered a living thing. So let's just review a little bit about what the scientific method is all about. It's basically just a process used to help solve problems, do experimentation, The major steps to the scientific method tend to be having a question or a problem, thinking about that question or problem, maybe doing a little bit of research about it to kind of understand a little bit more about it, coming up with a hypothesis about that question or problem, so basically coming up with a possible solution to it, and then testing and seeing if that solution works, making observations, then analyzing those observations, thinking about them, seeing did they work, did they not work, why not, and coming up with a conclusion regarding whether or not your solution worked or not, and why it didn't work or why it did work, and if more research or more experimentation is needed. So these are the relative steps. Do keep in mind that they do vary, and you know, you don't always have to follow these steps in the exact orders, and you know, every single step doesn't necessarily have to be followed, but it's just an overall process. Basically, you know, having a problem, trying to figure out a way to fix the problem, testing and trying to fix the problem, figuring out why it worked, didn't work, and continuing on and sharing your results, which can just be, you know, talking to a friend about it. So that's the scientific method. Microscopes, are basically equipment that is used in order to help view things that are too small to see with the naked eye. There are two main kinds of microscopes. Light microscopes, which are the ones that are allowing you to see living things because it uses light as its main method of viewing. You can view living things with it. You can also view non-living things, but light microscopes can allow you to see living, whereas the other one, electron, do not. Light microscopes do tend to be smaller, they tend to be cheaper, they do use light, but the magnification is less, only usually up to about 1,000 times magnification. We can see individual cells with this, but if you're trying to see like the parts of a cell, or even things smaller than a cell, like a virus or something like that, you're not going to be able to see it with a light microscope. Electron microscopes, which are much more expensive and larger, are going to be used to view things that need much more magnification. So we can actually use electron microscopes to see individual compounds, atoms, very, very tiny things, up to basically 1,000,000 times magnification. Unfortunately, with electron microscopes, you can only see non-living things, and that is because of the fact that it uses electrons that have to bounce off the object in order to be able to make the image. So that means the object has to be fixed, and in order to fix an object, you have to kill it. So that's the main downfall of an electron microscope. Again, also the fact that they are very large and expensive, so you're not going to see these in a school. They're going to be found in research facilities and labs. But now let's go back to cells. So now that we know about the cell theory, it's the basic unit of life, let's talk about the two major kinds of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells are simple cells that have no complex parts, including no nucleus. These are going to be things like bacteria, and actually that's the only cell that is a prokaryotic cell, are bacteria cells. They do not have any complex parts to them. They do not have a nucleus. They will have ribosomes. They will have DNA, but the DNA is just kind of floating around in the cell. They will have a cell membrane, and that is about it. Okay, not a whole lot more to them. Very, very simple. Pro means early. Okay, eukaryotic cells, you carry the nucleus. Eukaryotic, you carry the nucleus. These are the complex cells. These are the cells that have all the different parts to them. They are going to have a nucleus that surrounds the DNA and keeps it separate from the rest of the cell. They are going to have all kinds of different compartments that do specific jobs. All other cells, plant cells, animal cells, fungus cells, protist cells, all other cells except bacteria cells are eukaryotic. Okay, so only bacteria are prokaryotic, and simple. So here's just a little couple drawings just to show you the big differences between prokaryotic and eukaryotic. And this says prokaryotic animal cell, and that is actually, I'm sorry about that, that is a typo. It's not an animal cell because it's prokaryotic, so it can't be animal. So this is just a prokaryotic cell, and I wrote down here it is a bacteria cell. So you can see here that it's just got a cell membrane, which is that outer line. Inside the squiggly line is just its DNA. It does have some little hairs on the outside of the cell. Those are called cilia, which help with cell movement, and a flagellum, this little tail, helps move the cell around. But other than that, that's all that's in there. Occasionally you'll see some ribosomes, which are some little dots, and cytoplasm, which is just a filler. It's kind of like a jello that fills up the cell, but that's about it. Eukaryotic cells, and here is an animal cell that's eukaryotic. You can see there's a lot more going on inside the cell. There's lots of different parts in here. We've got the nucleus right here in the middle. You've got, and actually let me get, see if I can, see if I can point to some different things. Yeah, there we go. All right, so you can see my little hand now, hopefully. And so I'm pointing right now at the nucleus, which is in the middle. So it's got actually the DNA would be inside of that. Up here is what is known as a endoplasmic reticulum, and that's just a cell part that is going to have a main job of helping with making proteins and lipids and things like that. We've got these small little orange guys right here. These are considered mitochondria, which help with making cell energy, converting cell energy. Over here, these flattened stacks kind of look like pancakes that are stacked upon one another. That's the Golgi apparatus. They help with the transporting and packaging of the cell, kind of like a post office. So you can see lots of different things. There's the cell membrane as well. So much more inside and more complex in this kind of cell. Okay. So if we're looking at cells, if we're looking at a eukaryotic cell, keep in mind that they're kind of like mini humans. They do have lots of organelles, lots of miniature compartments inside that are going to have specific jobs. And so I've listed some here just to kind of review with you a little bit of what some of those parts are and what they do. So the nucleus is kind of like the brain of the cell. It controls what the cell does, and it's going to have the DNA in it. Cytoplasm is kind of like our blood as far as where most of the reactions and nutrients and things are going to be found. The cytoplasm of the cell is kind of jello-like and is mostly water, just like our blood is. Lysosomes are these little sacs that help to break apart things. It's kind of like our digestive system. They have enzymes in them that break things down. Lyse means to break apart. Mitochondria is the part of the cell that helps with converting energy, converting that glucose food energy into a usable energy the cell can use. We'll talk more about how that is done at another session. It's kind of like a battery, converts chemical energy into electrical energy or usable energy in a remote control for a TV or something like that. The Golgi apparatus, as I mentioned, is like the UPS, like the packaging and shipping part of the cell. The chromatin is the DNA. It's basically our blueprints, the genetic blueprints. The cell membrane is kind of like the outer skin or gate, protects the cell and controls what comes in and out. The cytoskeleton is kind of like the bones of the cell. It helps provide structure. Ribosomes, these are little miniature circles that are all over throughout the cell that help make proteins, since proteins are what actually make up the organism and do the work. And then I've got a few things here that are highlighted or starred, the cell wall and the chloroplast. These are both found in cells of fungus. I'm sorry, not those in fungus. Cell wall is found in fungus and in plants, and chloroplasts are found in plants. So these are not found in animal cells. Okay, but a cell wall is an extra outer protective wall outside of the cell membrane, kind of like an exoskeleton on an insect. It's just kind of like an outer armor or protection. It helps give structure and shape. Chloroplasts are what actually use the sun's energy to make food energy in plants. So plants are able to make their own glucose, and that is done with these specialized structures called chloroplast. Okay? So those are some of the parts inside cells. All right, now I want to take a closer look at the cell membrane. That is the main skin or gate that helps control what comes in and out of a cell. All cells have a cell membrane. And the controlling of what comes in and out of the cell is done via two different methods. Either the passive transport, where stuff just moves across the membrane on its own with no energy being used by the cell at all. No real effort by the cell. It's just allowed to come in and out through the pores of the membrane, basically because it's moving with the concentration gradient or going from high to low concentration. So think like a people mover or escalator. You just get on and it kind of moves with the flow. There's no extra energy by you or anybody else to make it move or go. It just moves. And that's going from one direction to the next. That's what passive transport is. That's things like diffusion, regular diffusion, like if you were to spray perfume on yourself and then in a few minutes the whole bathroom started to smell like the perfume. That's passive transport. It's just it's a natural movement of trying to spread out and equalize from that high concentration by you to the rest of the room. Another example is osmosis, which is basically diffusion but just the movement of water. Okay, water again is special and it is, has lots of unique characteristics, so it gets its own special name for its transport. So water is osmosis, the movement of water, and it moves in the same way from a high concentration to low concentration. Think of a water balloon. When you have all that water inside the balloon, and if you pop it, all of a sudden it gushes out, so it's moving from that high concentration inside the balloon out. Another example of passive transport is facilitated diffusion. This one tends to be a little bit confusing for students because there is the use of a protein in the cell membrane. However, the protein is not exerting any energy. It's basically just opening up a larger gap in the cell so that a bigger item can move through. So I like to explain it as if the person, if you were to go to a brick and mortar school and you had to actually walk to school, and we have the crosswalk person that holds a little stop sign on the road to make sure traffic doesn't come hitting you when you're crossing the road. That crosswalk person is kind of like the protein in facilitated diffusion. Okay, she holds the stop sign. She makes, kind of helping make sure that nothing happens while you're crossing the road, but you're moving on your own free will. She's not dragging you across the street. You're moving on your own, so there's no energy being exerted by her. So again, in facilitated diffusion, the protein's just kind of helping by opening up a larger channel. It's not forcing anything happening. So those are passive. No energy is used or needed. Active transports, on the other hand, active, think of hyperactivity, lots of energy. So active transports mean that the cell has to use energy in order to get things to move. It tends to move things against the natural current. Think if you're in a canoe and you're trying to go back upstream against the natural current. You got to put a lot of energy out there to make that canoe move up against the current. Okay, so active transports are kind of that way, and these also use proteins in a lot of ways, and the proteins tend to pump. They actually physically force the substance to move in or out. So there is an active energy movement being made, okay, with active transports. So here's an example again of the diffusion, like I was talking about. That's passive, natural movement, and I just have a picture of what that looks like if you were to, just drop some food coloring into a glass. Okay, you can see the glass is sitting here, it's empty. I dropped some food coloring into it and it's all concentrated, and so it's just without any energy being applied, no stirring, nothing, just on its own, it started to spread out and eventually equalize throughout the entire water. That was all done on its own, okay? And so that is no energy being used, completely passive. Osmosis was that other one. Remember, it was just the movement of water. Water has its special name. And I tried to show this using a little trick we can do with eggs. If you take some raw eggs and you let them sit in vinegar for a while, you'll dissolve the shell. Well, if you let it sit in the vinegar even longer after the shell has been dissolved, you'll notice that the egg will start to get very, very plump and almost like a big water balloon. And that is because the water in the vinegar tends to want to rush into the cell because the inside of the cell is not very filled with water at the time. So the water in the vinegar is going to tend to go inside the cell and make the cell plump up. So that is an example of how osmosis moves into the cell. Whereas if you were then to take that egg after letting it sit for a couple days in vinegar and stick it in Karo syrup, and for anybody that, if you know what Karo syrup is, it's basically where it's kind of like honey or liquid sugar, okay? And so obviously it's got a lot of sugar, very little liquid, so very little water. So you stick that egg in the Karo syrup for like a day or two, and you're going to notice, you can see down here at the lower picture, it's going to shrivel up. All the water that was in the egg is actually now rushed out, and that's because it's trying to equalize with the very sugary Karo syrup on the outside. Okay, and so it shrivels up, and if you feel it, so if you want to try this at home, you can, because if you feel the inside where the yolk was, it's going to feel kind of like a hard rock, and so you can see that all that water has kind of come out. So that's an example of osmosis moving out. And then here's the facilitated diffusion I was talking about with the crosswalk helper who helps hold the sign. You can see that this is a very magnified view of a cell membrane, okay? And so you can see the cell membrane is actually this kind of zipper-looking structure, okay? And this is a protein that's right here in the middle. And we're moving these large circles, not the small ones, but the open circles inside the cell. So this is the inside of the cell, this is the outside up here. And so we're just moving these, and there's a higher concentration. You can see there's a lot more of those open circles outside of the cell than inside. So it's going to want to try to equalize, and so those are going to try to move in. But they're a little large, so that's why there has to be a bigger opening than what is in between these other little parts of the cell membrane, and that's where the protein comes into play. No energy is used from the cell. The cell's not having to help it move in any way. It's just giving it a bigger channel, okay? And so again, this is still considered a passive transport. All right. Active, like I already talked about, and again, these do use proteins as well, but an active, remember, active actually pumps and uses physical energy, okay? So here you can see we're moving sodium, and in this case the sodium, that's what Na stands for, sodium. The sodium is getting pumped out against a current. Notice that there's already a lot of sodium outside of the cell and very little inside of the cell, so you would think the normal movement would be to try to get some of that from the outside in to try to equalize. But in this case, that's not what we want to have happen. So there is this forced movement of the sodium out, even though it's already got a lot outside, and that requires energy and requires a protein pump, which is going to physically actually kind of grab the sodium and force it out. So that's an example of active. We also have bulk movements, which are active, which require energy, and these are going to involve the Golgi apparatus, in which we're going to actually package things like hormones or waste and either bring them into the cell. As you can see here, endo means into, so endocytosis would be the packaging of something like maybe a bacterium that's immune cell's going to try to kill and bringing it in. Or if we're exiting, exo means to package and release, so here that's probably like waste or hormones that's being packaged and released. Both of these involve energy because of the needing to package and move like that, so they require cell energy. All right, And that is the end of session 1 and the review of basically the major compounds in water, the major matter that make up living things, as well as the cell theory and the different kinds of cells, the major parts of the cells, and then of course the cell membrane and how it works to move things in and out of the cell. So this has been the review for that, and in our next sessions we will be going over how the cell makes and converts energy. We'll also be talking about genetics and things like that. So please join and watch these videos for your review for your EOC. And if you have any questions about anything that has been discussed here, please feel free to email me and let me know, and I will definitely answer any questions you have. Please have a great day. Thank you for watching.